Hi everyone! This is going to be our first lecture on Chapter 1 in your textbook. So this is our general overview of genetics. So what we're going to just talk about briefly is what to expect for the rest of the semester, if you will. So this particular chapter is a really nice overview of what you're going to be learning this semester. So we're going to talk about a few things. We're going to talk about the different ways we can study genetics. We're going to talk about the molecule DNA. We'll talk about the differences between genes and alleles and chromosomes, and then we'll sort of finish off with some current applications of genetics. So let's first start with what is genetics? Well, in general, we think of the study of inherited traits and their variation as being the field of genetics. So when we want to know how traits get passed on from one generation to the next, and also what type of variation we see in those traits, we'll turn to genetics for those answers. It's a life science that should not be confused with genealogy. So we're not looking at ancestry here, so it's an interesting question as far as tracing your ancestors and where your grandparents or great-grandparents might have immigrated from at some point in history, but that's not what we're looking at. So we're not looking at the movement of people. That's kind of what genealogy is. What we're looking at is we're looking at the traits themselves that those people possess as they move from one location to the next. It's also an informational science that's having a huge societal impact. So there are more and more news stories out there lately about things we're learning about our genes and that information, what we can do with it, which is what we're going to talk about this semester as well. So what is a gene itself? If we're studying traits and we're talking about genetics, then let's define a gene. So a gene happens to be the instructions on how to make a protein. So if you didn't know it, your cells are basically made up of a bunch of different types of molecules, proteins and carbohydrates and lipids, and basically sugar molecules. And one important component there, one important molecule are these proteins. And these proteins are actually what our genes are coding for. And your genes are segments of DNA. And if you've heard deoxyribonucleic acid, that's technically what we call DNA, but let's shorten it and just call it DNA. So that's what your genes are composed of, these short segments of your overall DNA. And what's interesting is that the proteins that we're producing that provide us with these traits, so think of skin color and eye color and hair hair texture and a whole host of different other traits that we can both see and not see, sometimes the traits that we see are just a product of the proteins that your body is producing, and sometimes those traits are actually a product of both the protein you're producing and your environment. So that's where the study of genetics gets really interesting. It's not just the genes that we have that determine the traits we possess, but it's also an influence of the environment that we're in. All of the genetic information you contain is referred to as your genome, and it can be anybody's genome. So it's the human genome, that's all of our genetic information. We can also talk about the genomes of other living things. We can talk about bacterial genomes, we can talk about plant genomes, anything you can think of. So any living thing, which is going to be defined as essentially something that has a cell and can divide and has genetic information. All of those things have the same set of genetic instructions. We refer to these as the genome. So what's interesting about your genome is it's not just genes. It also happens to be really large sequences of DNA that don't code for anything. So as a matter of fact, only about 1.5% of our DNA actually codes for proteins. The rest of it, we're not exactly sure 100% what all of it does. We know some of that non-coding region of DNA is responsible for sort of turning genes on and off, so there's a regulatory mechanism there, but a lot of it is stuff that we are still not certain what purpose it holds. So when we're studying genetics, we can look at several different levels of genetic information being conveyed. So we can actually talk about things like DNA, which is the overall genetic sequence. genes, which again are those segments that code for proteins, and even chromosomes, which are like long segments of DNA, and we have a specific number of chromosomes in our cells. Humans have 46, as it turns out. So you can think of your DNA like a set of blueprints for your cell. Your DNA is the entire set of instructions on how to build and maintain a living thing. So again, doesn't matter what it is, it's a living thing. So your DNA is the set of instructions on how to build and maintain that living thing. And when we say maintain, what we're talking about is completing all of the chemistry that's going on inside of your cells. So the cells in your body are just basically bags of chemistry. And there's chemical reactions going on constantly inside of those bags. Your DNA, these genes are basically what's telling your body how to complete that chemistry, how to do those reactions, where those products are going, and what we're going to use them for. So here's kind of a nice diagram that your textbook uses. It takes your cell, so take a look at a cell right here, and let's say we cut it open, and we're going to learn about cells in the next chapter. We're going to cut the cell open, and we're going to take these long pieces of DNA called chromosomes, and if we unwind them, we get to the DNA itself, and then again, a specific segment of that DNA is referred to as a gene. So let's take a closer look at DNA. DNA is a molecule that's composed of nucleotides. So nucleotides are repeating what we call subunits of DNA. And each nucleotide consists of three things: a phosphate molecule, a sugar molecule, and one of four what we call nitrogen-containing bases. And these bases come in four different types: guanine, adenine, thymine, and cytosine. And we use the letters GATC to denote which base we're talking about. So if you've ever heard anyone talk about our DNA and kind of talked about it as a code, this is the code that they're talking about. They're talking about the sequence of letters, these adenine, guanine, thymine, and cytosine bases. This is the information that our cell is reading. You can think of it like a language that only uses four letters. And with these four letters, we can create all different types of proteins that our bodies need to function. So DNA is a double-stranded molecule. So you'll notice here in our diagram we have not just one set of letters, but we actually have two sets of letters. We're going to kind of talk more about complementary base pairing and how that all works in a later chapter. But in any case, that molecule itself is sort of twisted up, and we refer to the DNA molecule as a double helix. So again, your genes are specific sequences that are directing your cells to make certain proteins. And again, I said that your DNA contains both coding and non-coding regions. Okay, so coding regions are sort of what the gene is, and then the non-coding regions are the areas that are sort of turning on your genes off and on, like, yes, it's time to make this protein, or no, it's not time to make this protein. All right, so we're going to stop here and do a quick comprehension check. So I want you to think about the following question. Which of the following terms describes the complete set of genetic instructions or kits for of an organism? Is it a karyotype? Is it a single nucleotide polymorphism? Is it a gene pool, or is it a genome? All right, if you answered genome, that would be the correct answer. So the complete set of instructions on how to build and maintain something is referred to as a genome. All right, so here's another question for you. Which of the following do you think has the most genes? So we've got some E. coli bacteria here. We've got some a rice plant. We have a fruit fly. We have a mouse, and we have humans. So who has the most protein encoding genes? Okay, let's start with the smallest. Okay, E. coli only has about 4,400 genes. The fruit fly has about 13,000. Humans are next. We only have 23,000 genes, give or take. Mice have more genes than us. They have 29,000. And at the very, very top is rice. Rice has 50,000 genes. So we are an extremely packed organism, which means we put a lot of genetic information into a very small package. All right, so let's talk about various forms of genes. I'm not going to talk too much about alleles right now, but basically you can think of alleles as being two alternate forms of a gene. You get one copy of your genome from your mom and one copy from your dad. So each of those parents gave you a version of a gene that we refer to as an allele. Genotypes and phenotypes are just ways that we can describe individuals. We can describe their alleles, and we can actually describe the observable traits as well. So when we want to look at all of an individual's genetic information, we look at chromosomes, and I think I mentioned this very briefly, but humans have 46 chromosomes. You've got 23 from your mom and 23 from your dad. 22 of those are the same in both sexes, and then one pair of those are your sex chromosomes. It determines whether you're male or female. And then cells, I'm going to kind of skip over this, to be honest with you. We'll talk more about cells in our next chapter, but this is sort of just how living things are organized. I mentioned that your cells are made up of different types of what we call those molecules. Those make up different organelles, which make up cells, and then tissues, organs, organ systems, and organisms. So in any case, it all goes back to, again, these molecules that are being created, and your DNA is a big part of that. Genotypes and phenotypes we've already talked about. Traits can either be dominant or recessive, depending on how many copies of an allele that you need. And again, remember, this is just some basic introductory vocabulary. You don't need to know all of these terms right now. I just want to introduce them to you. We will learn all of these in other subsequent chapters that we're going to cover, but this is just sort of to give you an idea of the things we're going to be talking about. So we're going to talk about what it means to have a dominant trait or what it means to have a recessive trait. We're also going to look at how traits can be determined by not just the combination of alleles you have, but can also be determined by things like sometimes multiple genes interact with each other. Sometimes there's an interaction between your genes and the environment that you're actually in. So there are a lot of different ways that we can see how our traits are expressed, and we're going to learn about all of these during this course. So again, sometimes traits are just based on the genetics you have. So in picture A here, we have an individual who has a condition called polydactyly. Polydactyly means having extra digits, fingers, extra toes. And this is purely a genetic trait. It's something you either inherit from parents or you don't have it at all. Whereas something like skin color, for example, can change based on your interaction with the environment. So people with paler skin in the summer can sometimes have darker pigment produced when they spend a lot of time outside. So that's an interaction between their traits, their genes and their environment. So families are connected with diagrams called pedigrees. We'll take a look at what a pedigree is this semester. We will track different types of traits through generations, which are really interesting. Very quickly, I want to talk about populations and evolution, because after we have talked about all of these individual traits and how traits get passed on, we're going to relate that to how traits are tracked in populations and how these allele frequencies can change over time. So that's essentially what, again, our study of evolution is going to be in this class, is we're going to look at how these allele frequencies change in groups of living things over time. So we share an amazing amount of our DNA sequence with other living things, including chimpanzees, but we also share genes with all kinds of living things: mice, fish, fruit flies, yeast, and even bacteria. We have some genes in common. So there's a commonality in our DNA that is present throughout all living things that we'll talk about a little bit later on as well. So again, this is kind of a nice sort of little summary of the things that life shares. I'm going to kind of briefly talk about applications of genetics. So we're going to talk about DNA profiling. We're going to talk a little bit about genetics in medicine and healthcare. So again, I know I'm going through all that quickly. I'm going to skip this last question. Basically, which of the following is an application of genetics? It's all of the above. We can do all of these things. and then your review slide for this chapter, the important terms that you should know, and the important concepts.